think we have a decent amount of people in here. I want to say thank you to everybody coming along. I'm going to be talking about the LabChip GX2 Touch. This is a capillary electrophoresis system that we have placed at NBC. My name's James Geiger. I'm an application scientist with the LabChip, and I cover the region in California with the LabChip placed. And so also on this call is GXII Felwell. GXII is the account manager for the LabChip and also R&D scientist specialist extraordinaire as well. So we're both here on this call. And so it's a good opportunity to be able to speak to everybody and to be able to, meet everybody as well. So I'm just going to jump right in, give an overview for what the LabChip does, show you some example data, give you an idea of the different assays that can be run as well. I see we have something in chat. Oh, Jure saying hello. Awesome. Okay, so we'll jump straight in. So what the LabChip is capillary electrophoresis. And while I'm going to focus mostly on proteins today, most of what I'm going to say is completely true for DNA as well. And I'll talk a little bit about DNA. But traditionally, if we're going to do electrophoresis on protein, we would do something like SDS page, where we would take our proteins. These are folded and they have a bunch of charges, and we're going to denature them. We're going to essentially linearize the proteins, treat them with SDS, which is going to coat them with a constant molar ratio and give us a constant charge to mass ratio of these proteins. So effectively what we're doing through this is removing the influence of charge and structure, so that really the only thing that matters is the size or the length of this backbone of the protein. Then we take those proteins, we load them into a slab gel, run them, and we can see that the smallest proteins migrate very quickly and our largest migrate slowly. And we can separate these proteins based off of size. And this can allow us to characterize these protein samples, right? So we can know the relative somewhat purity of the protein. We can know the size of the protein we're worried about, the different moieties of protein that are there. Kind of issue with this old school method is that it is mostly qualitative. We're running like this, and we can get an idea of the size of each of these proteins and kind of the relative purity. But at the end of the day, it's just an estimate. We're just estimating roughly what we think. We could take an image of it and let a computer kind of look at the contrast between these lanes in the background and do some more calculations, but it's inherently qualitative. And it takes a long time to do, right? This is multiple hours of work. This is by the time you run the gel, stain it, destain it, we're at, half a day just to do, 10 samples. However, if we do the exact same thing, but instead of doing it on a slab gel, we do it on a capillary system, we can cut that time big time. So the LabChip is this system here that you see on the bottom right. And this is going to effectively run capillary gel electrophoresis. So the same type of thing that we're talking about with SDS page, pardon me, but instead of running through a slab gel, we're running through capillaries. And this allows us to run our sample through these capillaries, and we have a laser and a detector, and we do laser-induced fluorescence off of the proteins as they migrate through these capillaries. And that allows us to get what we call an electropharogram, which is real data. Instead of just qualitative bands that we're looking at, we're generating real data, fluorescence versus time. And that allows us to accurately quantitate the size, the concentration, the percent purity of each of these peaks. So the instrument here houses everything like the detector, we're looking at electropharograms on there, the lasers, electrodes. The capillaries are on these chips, which is kind of the hero of the whole system. So this is kind of a cartoon representation here, and I'll show in a little bit more detail what this looks like. But we effectively run electrophoresis separation based off of size of proteins and nucleic acids through these small capillaries, and that allows us to generate these electropharograms. Now, what I'm going to do here on these slides, sometimes I have an embedded video, but I think for purposes today, it's a little easier to just clip right here. And I'm going to show a little video that kind of shows what goes on during the run. And so our samples are prepared on a 96 well plate. And the first thing that's going to happen is we're going to bring the sample up into the chip. We're going to draw a vacuum from our big well on the top that we'll see right here. And this is going to sip the sample up into the chip, into those capillaries. Now, the amount that we're drawing is in the picoliter scale, and nanoliter scales what's going through these capillaries. So very, very small amounts needed. Once we bring up into the chip, we mix with a dye. So we're labeling the proteins like that. And now we have migration through some of these small capillaries that's powered through by electrokinetic forces and voltages in all these wells. At the end of the day, we're just going to pull a small plug of that sample down our separation channel. So we have this long separation channel down the middle of the chip. And once we're here in this separation channel, we're effectively doing electrophoresis. We're separating based off of size, and you see that in the video right there. Right before we get to our detector, we're actually going to do automatic destaining as well. So just like you would stain and destain your regular SDS page gel, we're going to do staining and destaining, but it's all automatic inside the chip. This gets rid of excess dye, excess SDS, better limits of detection, better quantitation. After that, we migrate through our laser and we do our laser-induced fluorescence. And that's going to show up in an electropharogram live on the screen of the instrument that looks something like this, where we see a fluorescence trace versus time. And so we can monitor it real time. And so I'll pause it right there. And so it's a fairly simple concept of what's going on here, right? We are doing electrophoresis just like you would do in an agarose gel or a polyacrylamide gel with SDS page, but we're doing it through these little capillaries. For our proteins, we have fluorescently labeled dye that's picking up, and the same thing for our DNA samples that we'll run through. The take-home message is that it is laser-induced fluorescence that's happening here. So this is going through a little bit of those destaining channels, a little more information on that big signal we get from all this free dye. After we destain, we only have signal from our protein, and that allows us to have robust quantitation, low limits of detection, all those good things that we like when we're doing any sort of analytical measurements. But it is laser-induced fluorescence off of a conjugated dye. And that's an excitation emission spectra of 635, 700. And so again, laser-induced fluorescence is our detection mechanism. So when we're detecting proteins, we're not detecting based off of A280 or any sort of amino acid profile of the protein. We should be detecting based off of this dye that's conjugated. And that's free of really the amino acid profile. It should be based on just backbone interactions with the protein. And so that kind of mitigates a lot of factors when we're quantifying proteins from other methods like extinction coefficients, things like that. We don't have to take those into account because we're not detecting based on that at all. Now, when we want to run on the LabChip, we essentially have a suite of assays that are going to come with reagents and chips. So for each assay you want to run, you need a chip and you need a reagent kit. In the reagent kits, we essentially give you everything you're going to need to run your proteins. So we have a dye that's going to be used to obviously detect your proteins, right? We have sample buffers that we recommend you use when we're going to denature your proteins. We can run reduced and non-reduced conditions. So what we'll do is take your favorite reducing agent, DTT, BME, TSEP. I've had people hand me 10x new page reducing buffer, right? Whatever you like to do to reduce your proteins, we'll spike that into this white tube here. And that can be used for reducing conditions. For non-reduced conditions, we can just run the buffer as is, or some people like to spike iodoacetamide or some sort of alkylating agent in there as well. But the gel ladders for sizing and concentration determination, internal controls, kind of an internal sanity check to make sure the instrument's operating well, wash buffers. The take-home message is that everything besides that reducing agent and your samples is here ready to go. So not much that you need to bring to the table from a reagent or chemical standpoint. Simply open the reagent kit and we're off to the races. The workflow for operating this is very simple. We have sample prep, we have chip prep, and these kind of go at the same time. We'll often, we'll start prepping the chip, start spinning down things, go prep some samples, go back and forth. But essentially, sample prep is the same as what you would do for SDS page. We take a couple microliters of sample, 2 microliters input, so very, very small amounts, which is awesome. We'll add that buffer to it, whether we want to be reducing, whether we want to be non-reducing, and we'll heat denature. So what's awesome is if we have a thermal cycler that can house these fully skirted plates that are compatible with the instrument, then we can just prep everything right in that plate, not have to pipette back and forth a bunch. But we denature these, we'll cook them at 70°C for 5 minutes, 80°C for 10, whatever works best for your proteins. We'll add a little bit of water, bring the volume up, dilute the protein down a little bit, and this goes into the instrument, just like we saw in that video. So very simple up here. The chip prep is also very easy. We'll take the gel in the red tube. We'll take the blue tube of dye. We'll mix those together. We'll spin it through a centrifuge to make sure we don't have any particulates, any microbubbles are gone. And essentially on the chip, you see we have these reservoirs up here. And what we're going to do is we're going to fill these reservoirs with our gel matrix. Here we have a diagram that's from one of our assay protocols. Really easy to follow. You're going to take the color-coded tube, take the green tube in the kit, add it to this well in this volume, 120 microliters. Take the blue dye that you've made, add it to those corresponding wells in this volume, and our destaining solution as well. And so we'll fill these reservoirs with the gel, and then the chip goes in the instrument as well. So all in all, when we do trainings, when we go slow, we talk through every step. This takes a little bit of time, but this takes 15-20 minutes of prep. Once you have the protocol down, you kind of know what to expect and know what you're doing. So really easy. And it's the same for DNA over down here. Sample prep's even easier for DNA. As long as your nucleic acids are within the concentration range, you just put them on the plate and you're good to go. So really, really easy for DNA. Now, the data you're going to get off of this instrument, this is an example of Protein Express. This is kind of our flagship assay. But this is an image of our reviewer software. And one thing that's cool about this, the reviewer software is totally free to download. There's a link we can provide. Everybody can download this on their computers. There's not any site licenses, nothing you need to worry about at all. So everybody can get this. You can get your data. You don't have to go to the instrument to analyze any data. Go sit at your desk with a cup of coffee and start looking through your results. So in this example, we're looking at our ladder. Okay. And what we see is our electropharogram. This is your real data that we measure, the fluorescence versus time, or in this situation, we've made the axis size, but essentially fluorescence versus time. Here we have our 6 ladder peaks. We take that same data, we represent it as an e-gel. So this is kind of a recapitulation, if you will, of this electropharogram up here. And so if you like to live in the old school and not look at peaks and look at gels, you can do that and compare your proteins that way. However, we also give tabular data. So based off of this ladder, we're going to calculate protein size. We're going to calculate protein concentration and percent purity. That's all automatically done in the software. So you should be able to open up a software and you can see for each peak, you can have size, concentration, percent purity, migration time. There's a lot of metrics you can add into these tables as well. I'll add, when I say that, people say, hey, do we have to control that? Do we have to play with it ourselves and manually integrate these things? You maintain full control over how it integrates, and you can change that as you need. But if you want just nice, easy, not having to do anything yourself, it's amenable to that as well. What I'm going to do now is show just a little bit of example data that we have from users using the LabChip. And then I'll go into a little bit more detail about the assays, kind of what to expect, time frame, how they operate, things like that. This is a nice little graph that I have from a group that was looking at different cell lines, different expression conditions for monoclonal antibodies. This was just a positive negative control sample that we set up, where we ran protein straight out of harvest, trying to look at which cell lines were going to express better this antibody of interest. And so what they were going to do with this is then start running this a lot. And obviously you would expect to see different cell lines produce different amounts of this 150 kilodalton protein here. So you can see all these host cell proteins down here. We can see well-expressing protein, poorly expressing protein. And then here's the gel representation here that correlates to these electropharograms. So you can see, oh yes, we have definitely way more of this antibody right here than we do in our in our negative control. Another example, another question that I get a lot is, we see a lot of pure proteins on our app notes and things like that, but can we run lysates? Can we run crude proteins? Can we quantify those things? And the answer is yes. It's very commonplace to quantify proteins really at any point in the production. So that could be whole cells, that could be lysates, that could be purified products. And this graph kind of speaks to that a little bit. It's similar to what we saw before, where there's 150 kilodalton protein that we care about, fluorescence versus time, right? And so we have insoluble, soluble, and purified fractions here that are all run on this protein. And so you can see here all this gunk in our soluble fraction, and then we have our protein of interest, and then we have our purified protein in blue. And the concentration, the percent purity of this protein of interest can be quantified all the way through, from how much goes into the soluble fraction, how much goes into the insoluble fraction, how much remains after purification, what are my yields like, what are my percent yields, am I purifying it really well, but am I losing half my protein? All these things can be quantified really nice and easily on the LabChip, all the way through. I haven't spoken so much to it yet. I'm kind of burying the lead of what makes the LabChip awesome, but notice the time scale here is only 30 seconds for each of these samples. So we're analyzing this. The end of this run is 42 seconds. So instead of getting a graph like this on a size exclusion column or SDS page that takes hours to run, we can do 42 seconds per sample on the LabChip. And that's going to allow us to do even a full plate, 96 wells, in only a little over an hour, hour and a half. Which is really awesome, allows you to process tons of samples quickly and, quite frankly, get better data than what you would get from kind of old school methodology as well. Another very common question I get is, what type of resolution can we expect on the LabChip? This is just an example of another kind of lysate sample that I've run with some users. And this shows the type of resolution that we expect to see on this Protein Express assay, where we're looking at these different proteins, a lot of different gunk, a lot of lysates of proteins in this lysate. But we can see between here, a couple kilodaltons, we do get pretty good resolution between these peaks. And so we should be able to get very similar resolution to other, electropharograms on the market, but better resolution between peaks, oftentimes from the SDS page shows that are being run. Many times we'll see, doublets appear when it's just kind of a blurred peak on SDS page, or a smear on SDS page. We can get a little bit better resolution there. Now, before I go into a bunch of assays on the protein side, I'm going to pause here. I'm just going to say, hey, I'm focusing on proteins in this talk, but everything I'm saying here is equally true about nucleic acids. And so I'm going to speak about more of these protein assays, but I do want to pause and say, hey, we can also run nucleic acids on this GX2 touch. And you can see the different assays that we have, same idea. We have a reagent kit and a chip for these assays, and DNA from 1,000 to 12,000. We can essentially quantify our nucleic acid fragments from 25 base pairs all the way up to 12,000 base pairs. We have NGS3K. This is a very popular assay that we have. This is looking at nucleic acid libraries before going into next generation sequencing. So doing a lot of smear analysis on these different fragments and things like that. But this is a very, very common assay. We have RNA assays as well, right? In your nanogram or picogram sensitivity flavors for whatever you need. And then small RNA as well, looking at only 20 to 150 nucleotides. The one thing I'm going to say here, just as a caveat, is these DNA assays are validated for double-stranded DNA. So double-stranded DNA is what we can quantify right now. Single-stranded, I always tell people, let's give it a shot, but it's really taking our RNA dye and throwing it in. So double-stranded DNA in any of these sizes, and then RNA as well. Okay, so getting in a little bit more to these different assays of what the LabChip can do. The take-home message here is that depending on your size of your protein, depending on the concentration of your protein, we have different assays for most of these. So Protein Express is kind of the workhorse assay. We have 3 assays that cover essentially from 5 to 2,000 nanograms per microliter. That is 0.005 mg per mil up to 2 mg per mil, linear concentration range. But if you have a protein or something where the protein is going to be very, very, very dilute, we do have a picoprotein assay that you can see our concentration goes all the way down. This assay is a bit different than our other assays. It involves off-chip labeling, so we do covalent linkage of our fluorescent dye to the protein instead of some of the methodology I showed before. But this allows us to pick up 50 picograms of protein. So essentially, if there's protein in your sample, the picoprotein assay is going to pick it up. And you can see that here. Now, again, let's say our typical workhorse assay has a sizing range of 14 to 200 kilodaltons. Well, if you have really low molecular weight samples, here we have a low molecular weight assay for you right there that goes all the way down to 4 kilodaltons on the low end. So essentially, it's just a matter of what's my concentration range, what's my size, and we can pick the right assay there and get data just like we saw before. Now, to talk through just a couple of these and kind of show the difference with what we're talking about, we'll do a little compare and contrast here. So Protein Express is the one that I've been showing a lot of data on. Again, this is kind of the workhorse assay that most people are running. This assay is going to give you size, concentration, and percent purity of your proteins. It has a molecular weight range of 14 to 200 kilodaltons. Your runtime per sample is 42 seconds. And these chips can be used 400 times. This is a really good thing that I want to highlight here, is that this isn't a situation where you use this chip, you have to run all those samples at once. You can either do a 384 well plate and do the entire lifetime of this chip in one run. You can do 20 samples for 20 days and keep reusing that chip over and over again. It's totally up to you, but these chips are reusable. That's not something that's super common in this chip-based detection field. And so really nice to have a reusable chip here. It's also not going to lock you out at 400 samples. So if you load a chip in there that's run more than 400 samples, the system's going to go, hey man, this chip has more samples than we recommend on there. Do you want to keep going? And you say, yes, I want to keep going. So we don't lock you out. You do maintain control. I always just tell people, if you're at your 2000 sample on a protein chip and you send me that data, just know the first thing I'm going to tell you is to try a new chip. But we also have, again, here, our low molecular weight assay. It's pretty easy to see what's going on here, right? The same exact idea, but we're doing a lower molecular weight range. A little bit longer runtime, same validated amounts per chip. For proteins below 14 kilodaltons, do we have proteins in this size range? Maybe we have proteins at the 50 kilodalton size range, but maybe we want a little better resolution in that 5 to 80 kilodalton range. Sometimes the low molecular weight assay can just give you a little bit better separation than Protein Express in that same range. And then our picoprotein assay, very low concentrations, picogram quantities of protein we can pick up here. So it's just, again, about picking the right assay for the proteins that you have in your project. On this, I get a question about sizing quite a bit on the top end. So here's an example again of our ladder where we run from 14. We have 120 kilodalton peak here. We validate the extrapolation of this linear regression of our ladder peaks out to 200 kilodaltons. But you'll notice that there is data collected out past the 200 kilodalton range. And we do allow users to, in the software, you could spike in your protein into our ladders, maybe add a peak out here. That's going to give you a little bit better regression, give you data out a little bit further. So there is some flexibility to do that. It's not something we really validate. It's kind of on users to kind of internally validate it themselves, but it is an option in the software where you can extend that range and get some data past that 200 kilodalton range that I showed here. Okay. We have a couple other assays on the protein side for CESDS that I want to talk about. And we're comparing them again to Protein Express, this kind of workhorse flagship assay. And these are our latest generation called IntelliChip assays. These are protein clear and protein exact. And what essentially these are is, next generation assays that involve smart technology and essentially internal calibrations to ensure that we have more robust statistics, more accurate quantitation. And I'll talk through a little bit of that in just a second, give you an example. So protein clear and protein exact, you can think of protein clear as an assay designed specifically to tell you the percent purity of your proteins. So if we need to know percent purity and we want really accurate, robust quantitation, protein clear is the default assay that we choose to go to. There's two-step calibration. I'll talk through that again in just a second. Protein exact is an assay that is, you can think of it as just the next generation of protein express, right? It's going to give you size, it's going to give you concentration, percent purity, but it involves these internal calibration steps that give us better reproducibility, better quantitation. And so one area that this is commonly used in is quantitation of AAVs in cell and gene therapy. And so a lot of these things, labs producing AAVs, these are GMP facilities, they want quantitation, they need really good accuracy, they need really good comparison between multiple instruments, between multiple projects. And so protein exact is a really great option for them. You can see we have a little less throughput here. We run 96 samples in a plate as opposed to 384. These do come with high resolution chips, and this is a little bit less or longer runtime, but still only 65 seconds per sample. You can see we have a slightly larger molecular weight range here. So again, as I talked about, you could quantify off here. We have the ability to spike in things of the ladder, but if you have a protein over 200 kilodaltons, then one of these may be a good option as well. So essentially the difference between how protein express works and how protein clear and protein exact work is this internal calibration step. Most of your prep is going to be very similar to actually run this assay, but the system itself and the software is going to calibrate to make sure that your data is very consistent. And so let me just show you an example. This is protein clear, and no need to, we're not going to talk super in depth on this, but effectively what's going to happen is before we run samples, we're going to run through an algorithm with a known standard. In protein clear, we have our Verimab standard that we send, and we have a known expected result, a percent purity of these two peaks in the sample that we expect to see. And so we'll warm everything up, we'll get everything ready for the run, we'll run this standard. We'll see, hey, test, did this come up with the actual thing that we want to see? So we run 5 different tests, right? Different voltages down the channel, different destaining currents, these types of things. Hey, are our results good? Is this what we expect? Yes. Okay, we're going to run through the sample with making sure with the proper voltages and what gave us the best result. No, are we not happy with that? We're going to go back up here and iterate, and we're going to make sure that we get that result off our known standard before we even put your samples onto the chip. So there's this internal calibration, this algorithm that we run through to make sure that everything's reproducible and that the results are as expected before we even put your samples on. The results of that are this. And so this is an example still from protein clear, but this is showing intra and inter-run reproducibility. So when this graph, this is from an app note for protein clear that I'm showing. This graph was run, I believe, on 2 different instruments, 2 different people. This is an overlay of 5 runs of this antibody. And it was 2 different chips, 2 different people, 2 different instruments, 5 runs. And you see really, really good overlay of this data. So this helps boost our reproducibility, regardless of the instrument that's being run on, regardless of the person prepping the gel, prepping the chip. Really awesome that we can have really good reproducibility like this across these different scenarios with protein clear. And the same goes for protein exact. These internal calibrations allow us to achieve better statistics, a little better resolution as well. Okay, so that's a lot, but I'm going to highlight 2 other little assays real quick that we have. So what I've talked about before is our CESDS offering. So looking at size, concentration, percent purity of proteins. And then I talked again a little bit about nucleic acids. What I want to talk about here is 2 additional assays that we have on the protein side. And the first one is charge variant assay. This is capillary zone electrophoresis. And so what this is meant for is to separate your proteins based on their acidic and basic moieties. And so this is going to essentially separate based on the PI of these different moieties. So what we do is we select a running buffer to fill the chip with at a certain pH. Add your samples into the plate. We'll label your samples with the dye that's provided, again, with a reagent kit and a chip for this assay, just like the other ones. Those are then loaded onto the instrument and we run. And we're still in the range of about 60 seconds. There's a few different options you can choose. You can do a long assay to let it migrate longer if you need to, but we can address that when we actually do this. But essentially what's going to happen is certain moieties of your protein may be acidic, right, in that pH. So those side chains of these amino acids are going to be, they'll have hydrogens on them, or maybe they won't. Those acidic variants will migrate quicker because they're drawn towards that negative electrode at the end of the capillary. The main variant will migrate a little slower. And the basic variant's actually drawn the other way, not moving through the channel as quickly as everything else. And so what this does is to take these nice clean peaks that we get on CESDS and it stretches them out, and we can quantify the amount of main variant that we have versus acidic or basic variants in our sample. So this charge variant profile, this kind of fingerprint, if you will, can be a key quality attribute for things going out the door, like in therapeutics, something that just needs to be quantified, make sure we have the same range. Also, it can be very helpful. I've seen people use this for making decisions about leading candidates, right? Or you know, why is this protein not behaving as well as it should? I've seen people run this assay, and what they're testing is how this variant profile looks when they elute in different pHs, when they elute with different buffers, things like that. And we can see that everything looks the same on a gel, but when we run this, then, oh yeah, I have double the acidic variance here, and I'm changing this profile of what the protein looks like when I put it in a different buffer. And so it can be a nice way to do kind of a QA, QC, even in the process of developing these therapeutics as well. As an example, this kind of talks about what I just showed. This is a nice monoclonal antibody here. And when we do CESDS, this is a denaturing assay, right? So we get this nice, beautiful peak here at 150 kilodaltons, and we have some moieties down here as well, some impurities as well, but we have this nice, beautiful peak. And so then we take this, and when we run a charge variant assay, we're not denaturing it, it's still folded. And we can see that while this looks like a beautiful peak, only about 25% of that is that main variant of this charge profile. We have a lot of different basic variants. We have a lot of different acidic variants. This is actually data that I generated with some people on site that they were looking at, again, different elution conditions. We could see that these basic variants move up or move down depending on their elution buffers of choice and how they were handling these proteins. And that actually correlated pretty well to what was the leading candidate, why some were behaving really well, why some were doing not so well in their activity assays. And so this was actually a really powerful tool for them to be able to make decisions about what they were going to move forward with for their SOPs and protocols. Okay, last assay I'm going to highlight here is our glycan assay. This is a super, super cool assay where essentially what we're going to look at is N-linked glycans that are bound to proteins. And so same idea, we have a chip, we have a reagent kit. And what we do is provide everything you need to go from your Eppendorf tube of your protein to looking at a trace like this, where we can look at our glycans in about 4 hours time. And what this entails is taking your protein sample, and we'll denature them on the plate, cook them at 70 C for 10 minutes. Then we'll add in our enzyme, and we'll do enzymatic cleavage of these glycans, where we'll get the glycans free in solution off the proteins. We'll take a little bit of those free glycans. We will add that to our dye that's going to label these glycans. We'll let these incubate for a couple hours at 55 C. We'll reconstitute them in water because we actually do an evaporation reaction here, and then load the plate directly on the LabChip. And then once we're on the LabChip, we're looking again, we can see our scale of 40 seconds per run total. And what this is going to do is give us migration of these different glycan moieties. And so here we have these 6 main glycans for IgG molecules that we care about mostly. And we'll take our standards. We can look at the runtime of these standards and compare to our protein of interest. And we can quantify relative abundance of these different glycan moieties. I think this is a super awesome assay because a lot of users are having to ship off proteins to a different company, or even if you're in the same company, you're shipping it to a different lab, a different site, waiting for weeks to get your glycan data as they run, LCMS or something like that. Whereas this, on the same instrument that you're quantifying your proteins on, you can get this glycan analysis in a few hours, same day. It's not rare when we do demos for us to do glycan analysis and CESTS in the same day on the same samples. And so that's a really powerful tool to be able to quantify these things. Here's an example of a little bit more of a complicated glycan run where we were quantifying charged sialylated glycans, not just those nice, easy 6 glycans that we showed before. This is our extended range glycan assay. And we have an app note going into more detail on this as well. But the take home message is we can quantify some pretty complex glycan traces and fingerprints on this instrument. And we have, again, an app note on this. We have dilution studies of different concentrations and how things don't really change with migration times, don't change with dilution, things like that. So it's not just these really pretty glycans that we can do. We can do these more complex patterns as well, from IgMs, more other proteins besides antibodies, right? So we have IgGs here, but anything that's N-link glycosylated, we can get nice patterns just like this. This slide just shows a little bit of information on why you might want to do this, right? We see different glycosylation patterns as a function of the clones that are being used to generate the proteins, different constructs, and then different expression conditions can lead to different glycosylation patterns as well. And these glycosylation patterns and how much of each glycan is there can affect the protein's ability to get where it needs to go in the cell. It can affect the protein's ability to actually do its job and bind what it's supposed to bind. So another one of the key quality attributes that needs to be quantified on a lot of biotherapeutics that are going out the door. Again, really rapid, really easy, really awesome to use. So that is the last assay that I wanted to talk through. I know that was a ton of information. But the take-home message here is that nucleic acids, protein, size, concentration, percent purity, charge variance, glycosylation, these are all really easily and robustly quantified on this LabChip platform, and in an amount of time that's really unparalleled in the field. 60 seconds per sample, you can do 96 samples in an hour and a half and have really robust quantitative data across all of these assays. And so really a game changer if you need the throughput, and really a game changer if you're using SDS Page or any of these older methods, because you can get better quantitative data based off of this. That's the last slide that I have prepared here. But what I want to show, just to give a little bit more detail that we can provide as well, is part numbers and chips for each assay. So again, we have serial numbers for our reagent kit and for our chip. We can see for each assay that we're interested in, we have a chip, we have a reagent kit. And so we can get each of these and kind of know which one we need for each of our protein, like I mentioned before. With that being said, the last thing that I kind of want to highlight here is that we do, Jure and myself, want to have time to come in and do trainings. If anybody's interested on learning how to operate the system, we can run some samples with you as well, kind of show you how to do data analysis, how to get the best use out of these assays and the best pro tips using the instrument. And so with this in mind, and these different serial numbers and reagent kits and different things, we do have this little Excel sheet that we wanted to show. And so this is just in the next week, from the 28th to the 5th. We can also be flexible moving forward as well. But just in the next week, what we want to make available is, if anybody's interested in this, we want you guys to be able to sign up for different slots. We could do a couple in one day. But we'd just like to know, who you are, the users that we will be training, and then the type of assay you're interested in. the preference, sample type. If you're not sure of the assay preference, then you can type in, I have a protein of this size and this concentration. You can fill all that in right here. And then we can make our recommendation based off of this right here. But if you know already, based on kind of what we talked through, or available resources online, then you can just fill that in right there and we know as well. And so with that being said, I've probably been talking long enough. I'm sure everyone's sick of hearing me talk. I do want to pause and see if there's any questions from anybody. Anything I can go back through those slides and kind of rehash some of these things if we need to, but I'll stop. And Jeray just shared that link to sign up for different training sessions in the chat as well. So there's the link to this Excel sheet right here. So I'll be quiet now, and I want to open this up to any questions for anybody who may have anything that they want to say.
I don't have a question, but I would say thank you so much, James. It was a great presentation.
Always a pleasure, man.
Hey James, this is Avi with BioLegend. Maybe I'll break the ice with
Cool.
a couple questions here. Great presentation. Really appreciated the orientation. It's kind of a basic question. If you're running protein assays versus DNA assays, does that use a different chip? It looks like you use different reagent kits, but do you use different chips for those?
Super good question. So essentially what I recommend to people, like you can see here, chip ID for this assay and this assay are the same, right? So some chips can run, so protein assays and DNA assays, there we go. Don't want to click on that link. Some protein assays and DNA assays are going to use different chips. Let me scroll back up to that DNA table that I showed before. You can see that this same chip is used for a lot of these different assays on the DNA side.
Oh yeah.
So,
okay.
So what we typically recommend, just to be honest, just to not, cross too much, is typically get one chip for each assay type. And I'll normally write in a Sharpie, hey, even if this chip can be run on multiple assays, I'll say, hey, I'm using this chip for this assay. On the DNA side, that's less of a issue, but on the protein side, for instance, I probably don't want to run proteins on the chip and then glycans the same day. There may be some residual kind of stuff inside that chip. It does wash out really well, but we kind of do open up ourselves to, I don't want residual glycans hanging out in that capillary when I'm trying to run my proteins through, right?
Yeah.
So typically, there is a little bit of one chip can be used for multiple assays, but I like to kind of keep them separate just as my own personal way of doing things.
Nice. And then if I could get you with a quick follow-up question.
Yeah.
If you're doing like antibody analysis, and it was good looking at those clean 150 kilodalton peaks, are you able to run reducing conditions? Yeah. And does that happen? Do you kind of do that in sample prep before you load?
Yep, exactly. So here's an example here, right, where we looked at these ones that are pretty clear. But yeah, so what you would do is when you were to prepare your buffer here, so you would take a little bit of protein, a little bit of buffer, mix it, and put it in the well before we denature. You would just spike in whatever you want to spike in for your reducing samples. So let's just say DTT. And you can prep that in the same plate. So you could do I'll have users do a full plate of non-reducing and then a full plate of reducing. I'll have people that'll split it right down this way on the plate, where this half is non-reducing, this half is reducing. I'll have people that load them side by side. Reducing, non-reducing, reducing, non-reducing, reducing, non. And then you can side by side see each way that you ran your protein. So there's really a lot of flexibility in how you want to run it. It's just a matter of putting that chemical into this well with your buffer when you prepare your samples.
Beauty. Thanks.
Yeah, definitely. It's a great question.
Hey, James, this is Serena. Nice job. I had a question from like office hours. I heard like, how do you clean the chip and how do you properly store it so that you can use it for 400 runs? Like, is there a way to improperly store so that you get smaller amount of runs out of it?
Yep. Great question. So I'm going to say this on the DNA side, and then I'll say it on the protein side. So what we have, this kind of shows it right here, is, and in each of our protocols, in fact, here, let me just pull this up real quick. Give me 1 second. I think I can pull it up if I do this. I believe it shows it actually in our protocols, in our big user manual. Preparation procedures. So what we'll do is we typically recommend you have a vacuum aspirator because it makes it nice and easy to do. But for cleaning the chip, what we'll do is just take some Millicue water and we will fill up the wells of the chip with Millicue water and aspirate it twice. That's essentially all we do for cleaning. Now, if you're on a DNA chip, if you read through this protocol, it'll say, okay, we actually provide you with a washing storage buffer in the kit. You can take that storage buffer, fill up the wells with the chip, and then you can put it back into the instrument and hit the wash button, and that'll flush all the capillaries out. And then you take the chip out, put it in its case, and that can be stored at 4C, totally good to go. Okay. For the protein assays, we don't actually recommend you do that wash step. You don't have to. You just take the chip out of the instrument, you'll fill the wells, aspirate with water, all the active wells, and we just fill them back up with the Millicue water and store them. The big take home is that we don't want these wells to be dry on the chip. We want them to always have liquid in them. And then a protein chip, we can actually store at room temperature after we use it. We actually kind of recommend after you use a protein chip to not put it back in the fridge, just store it in a bench, maybe in a drawer right there. We run a bunch of SDS through these capillaries, and if we put it back in the fridge, we kind of can have a situation where it's possible that the SDS could crash out in those capillaries, and we don't really want that, obviously. If it happens, we can mitigate it. We can get the chip functioning again. But probably the best thing for protein chips would be to just follow this, aspirate them with water, make sure we're doing this before and after each run, and then we can store them at room temperature to make sure none of that happens that I mentioned. But that's a very good question, Serena. Thank you for bringing that up.
Thank you.
Well, if nobody else has any questions, I will stop recording the video, and I would like to thank you all, PerkinElmer and all the attendees here for the seminar, for the webinar, and we are looking forward for the next one.
Awesome.
Thank you very much, Flavia.
Thank you. Thank you, Serena. Thank you, James. Thank you, Dre.
Thanks, everyone.